Thank you, Nick. Good afternoon to everybody. Thanks to the organizers, both Mike and also Tecan in Future Diagnostics for having me here. So I have to hijack you now for some time because it's not on clinical tests. It's more on philosophies, how to try to get new biomarkers discovered and then how to move them forward. I had to skip some of my 800 slides because we're a bit running late. But let's see how we will come forward. Is this one working? It's not working. It's working as a pointer, but not to advance the slides. Okay. Also, the website is given. If there's any interest, please contact me via the website. Autoimmune diseases is the focus we are working on. There are more than 80 autoimmune diseases, some with very strange names based on the persons that discovered the disease or on the first patients seen. Interestingly, they are the second, one of the top 10 leading causes of death, and right now they are the second biggest area where drug development takes place. After oncology, it's no longer cardiovascular and metabolics, but it's autoimmune diseases, probably given to the fact that cure is impossible right now. There's no cure for any autoimmune disease. They are chronic, they are disabling, so they are also very much on the rise in industrialized countries. So pharma companies have learned that monoclonal autoantibody therapy in autoimmune diseases is quite good business, and there's big medical need. One thing which is quite important, you see down here, average non-responder rate to any kind of medication is 50% or higher. So if you have any autoimmune disease, you go to your doctor, even if he's best in class, the chance that he will prescribe a good medication for you is 50% or less. In 2015, that's a rather sad statement, but it's fact. That the reasons are a little bit depicted on the spidergram, where you see you have joint stiffness, you have pain, blindness, myocarditis, skin rash, fatigue, numbness, seizure, many different organ systems involved, inflammation, pain, many of these overlapping syndromes, at least in the first parts of disease, hinder early diagnosis. And early diagnosis would be a cornerstone because then you can stop disease at a certain stage where long-term effects are not as prominent. More than 50 million patients in the US alone, so it's a huge market and it's underserved both in terms of diagnosis and in terms of medicine. The testing population we have to look at typically has a year-long course of disease. There's a preclinical phase where patients just have mild symptoms and they may go to their general practitioner for some whatever symptomatic treatment, but it's not really recognized as a certain disease. They're typically undulating diseases, times of relapse, times of remission, patient feeling better than getting progredient symptoms. Genetic environmental factors are part of it, and one of the hallstones of diagnosing them is autoantibodies, autoaggression. So the immune system is overactive, not like in oncology or infections where it may be too weak to combat the disease. It is overactive and starts to be self-destructive. At that stage, one would love to have a clear diagnosis and therapeutic alternatives. Most often they are like at stage 2 or stage 3 when the final diagnosis is made. The spectrum of diseases are known to most: rheumatoid arthritis, systemic lupus, and many of the others. If we now look at the type of analytes which are quite prominent, it's human IgG. It's the main force that drives the disease, that attacks internal tissues, organ structures. It's also a very nice and elegant analyte. It has been used for decades in indirect immunofluorescence. We just can take patient serum, put it on a tissue slide or on a slide expressing whatever proteins, and then you can say, okay, this patient has an autoimmune disease because it's reacting. That does not really tell you what is the target of the autoantibody, but research over the last 20, 30 years has brought many targets, as you can see in that list, which is also on the PDF that you have on your sticks. So I don't need to go through all of them. It just shows you that the many diseases do share and not share many of the autoantibodies. And one specific thing which makes it different from typical diagnostics, a patient may have a disease but lacks the autoantibody, and so that a missing of an autoantibody does not exclude the disease. You can only include the disease by measuring the autoantibody. You cannot say it's not the disease if you don't have it. Most of those diseases do not have 100% coverage in autoantibodies yet. Most often at late stage of disease, when the clinician knows what the disease is and does not need IVD, they come up to 70–80% of coverage. In early stages of disease, you may only find 30% of patients having these autoantibodies. And whether this is due to the course of disease or whether our discovery efforts haven't been good enough or whether our sensitivity in existing assays is not good enough remains to be seen. We don't know yet. That sets the stage for our interest in autoimmune diseases. There should be more autoantibodies available and other biomarkers. If we look at a bit older slide from 2002, introduction of new clinical biomarkers is slowing. New approved clear diagnostic protein tests declined in the last decade, from 1992 to 2004. Basically, it went down to one test a year, which is not really good enough to serve the medical needs that we all have. Then we have seen the advent of those different omics technologies. In the mid-90s, it started with proteomics and lipidomics and glycomics and economics. No, that's a different one. So all these omics type of technologies where you can look at hundreds and thousands of analytes with the hope that this modern technology will give novel biomarkers to help science and to support medicine. And then that's just a sketch for a typical proteomics type of study. With today's mass spectrometers, which are wonderful instruments, you can take a serum sample, a tissue biopsy, and get far more than 1,000 proteins or peptides analyzed from every single sample. That is very good. It's giving you a comprehensive analyte picture. Most often in literature, the study size of publications is 10 to 50 samples because it's so much work. I put 3 question marks in here. Most often 3 to 10% in all these papers seem to be differentially regulated marker candidates. Maybe that is owing to P of 0.05, and biologists love to make a cut at that stage. A good point is that we get increasing sequence coverage of candidates. The times that you had spots on the 2D gel are long over. You now know what are the differentially regulated ones. We even have advances in quantitative mass spectrometry, that you get quantitative differences by isotope dilution, mass spectrometry methods, and that, which is certainly is good, rather better than qualitative information. And every paper, at least in the discussion later, has the saying that they all have to say there's a high degree of biological diversity. If they work in cell lines, it's easy. If they work in monoclonal whatever or clonal mouse systems, it seems to be easy. As soon as they go in humans, it's very diverse. The disease is diverse, the persons are diverse, it's lots of environmental factor adds to diversity. What also is of concern, if you look at pre-analytical plus technical variation in these omics papers, if they state them, it's typically CVs of 30, 50, 100%. And coming from an IVD perspective, that is no good assays. So you look at thousands of analytes, and every single data point is of weak performance, 3-, 4-, 5-, so that's not really good enough. High false discovery rate, lots of what is published will not be reproduced. Poor repeatability, need of extensive validation work. And need of extensive validation work means labor, costs, time. And unfortunately, the scientific community is not incentivized for that. If you take all these points, those pluses for yourself, you can make papers with high impact points on 50 patients. If you then go and try to validate what you have found, you need 500 or 1,000 patients. It takes 4 years longer to do all that work. It's very expensive, and if you publish it, it will not give you lots of impact points. So there's a disincentivation for validation work in the scientific community. And now look what happened in the years after 2002. Yeah, we had a peak year with 4 new markers, okay? But until 2009, it's 1 or 2 and maybe 3 novel proteins. So it has not really changed. So the dilemma that we have too few good markers introduced into the clinical system remains. And if we compare biomarker development with drug development, it's also a phased process from targets and validation work through to different phases, marker concept, validated disease association, and so on. But here you have a black hole. That's a black hole of marker validation. So the take of it is that you have lots of front loading here and just a trickle coming out there. And as a biotech company dedicated to omics type biomarker work, we were thinking on how can we combat that. So the first plan we had was we need a high performance omics discovery of novel autoantibodies, meaning every single data point needs to be as good as possible, even being less comprehensive maybe, but skip those that don't perform well in order to concentrate on those that have good discovery assay performance. Harvest through many autoimmune diseases because they are all kind of intermingled and mixed. If you just find one marker in systemic sclerosis and you have not screened all the other diseases, it may turn out later that it's unspecific, so you won't gain. Focus on some, and for the company, forward integration, that's not the topic for today. Going into the technology that we've set up, we were fortunate that at that stage, a bead-based technology from Luminex extended their frame. Luminex works with color-coded beads. They now have 500 different colors in those beads, which means that if you couple a protein as an antigen on a bead, the bead color will always tell you what was the protein of interest. So you can couple up to 500 different proteins on 500 different beads, indicated by the different colors here. And then you can create a bead mix, which means it's a multiplex test. It's an array, a bead-based array. They run nicely in 96 well plates, so you can run many, many patient samples with 500 antigens per assay, put that through the machine, and get your autoantibody response. Over the years, we were quite busy producing more than 7,000 human proteins, putting them on beads and screening them. So for a single patient, we needed to do 14, 15 bead mixes. But since you only need 1, 2, 3 microliters per serum per assay, it's not too heavy on patient sample consumption. So that's what we call Sero Tag Platform. We have now analyzed a good number of patients, 7,000 human proteins, 5 microliters required. We look at IgG, normally all isoforms. We could look at IgM, IgA, but focus is IgG. The nice thing is that the Luminex system is widely used and accepted by FDA, EMA, and all the others. So we have a discovery technology that could be the IVD technology in the later place. So you would not need to change the platform. So over the years, we have now consumed some 85 billion beads. We didn't count them one-on-one, but it's an average what we bought from Luminex. They're quite expensive. Nice is they are magnetic, so typical kind of automation procedures work fine. And we have analyzed more than 10,000 patients in these diseases, which means that any antigen that we put in, we have 10,000 data points in healthy, in old, in young, in female, in male, in black and white, and whatever. So we think it's a quite comprehensive database that gives us a good grip on an individual antigen to say, are you worth further efforts? This instrument is from a Swiss company hosting us here, Evo 1500 with whatever carousels. So automation is an important part. This is from a medium-sized screen, the microtiter plates. That type of raw data output then undergoes distinct bioinformatical procedures. Per run, we can have from 8 to 1,400 sera, so we can really do discovery on a framework in proteomics that was not possible before. The typical performance is about 5% repeatability, which is quite nice. It's quite already close to clinical IVD, and we can start. We can use a single antigen or even 7,000 antigens, and if capacity allows, we will extend that by screening literature for novel antigens. Now going on to a case, systemic lupus challenge. That's really a beast of disease. The pathogenesis is very complicated. If you have 4 out of 11 different symptoms, then you're called a lupus patient. Any 4 out of 11. It affects multiple organs. There are differences between ethnic groups. It is remitting and relapsing, so if you have a clinical trial, your placebo group will typically have 25–30% of response. So your novel drug has really a tough time to combat that 25–30% if your expectation is that your drug will only be 50% effective. That creates trial sizes of 800, 1,500 patients, trial durations of 5 years for phase 3 trials. So very expensive, very long. Over the last 50 years, 1 novel drug was approved by FDA for treating lupus. So that's really a nightmare. Reimbursement for multiparameter assays remains a challenge, which means that in such a complex disease with so many organs, disease activity scores and whatever, if you want to measure cytokines plus DNA, RNA plus inflammation plus, very quickly the reimbursement system says, no, we won't pay for it. So outside of this presentation, that is something we really need to be concerned about. And diagnosis, differential diagnosis, biomarker support for drug development, companion diagnostics are all medical and industry and society needs. So first we did is that we took known antigens, so the old guys, the good guys, took a 100 patient cohort where we had a clinical readout of different antigens that are common in that stage. We used our proteins on beads and also many other ones and tried to find out, can we match what is given from the clinical IVD type of site. And if we look at like SSA, 42% were positive and the clinic said 37. So give and take, we were in the same ballpark, validating that our system works, even if it's just 500 proteins in one Eppendorf cup. So that gave us enough fodder to say, let's tackle that. Starting discovery, meaning taking a fish trawler with a big net and going through the ocean of lupus biomarkers, try to take everyone who was above a certain scientific threshold and statistical threshold, accepting a false discovery rate, which is rather large, because we knew that after the discovery study, we have to go through the same expedition several times. Replication verification studies, so we did 3 studies and then concentrated on those that robustly popped up every time. Came back with a robust portfolio of about 50 novel SLE antigens. Then this is an organ registrar where you can pull and put back on the different registers, where you then can say, are there some that are linked with nephritis, yes or no? Are there some that are linked with disease activity, yes or no? So that we could create different panels of 1 to 18 markers. And also could look at multiple applications. So the top panel would be prognosis, the bottom panel would be response prediction. So we would really try to harvest through that as good as possible. And then coming to prototype assays, medical claims, design goals, and so on. That puts it together in a slight time frame. In Q3 2012, we did a discovery study with 6,000 human antigens, 100 SLE patients, and we had healthy control and also active controls, RA and systemic sclerosis. We replicated that with those candidates that looked best, and we added a working cohort from workers at a large German company that got offers of a rheumatoid arthritis screening. We did a verification study, again using now 7,000 antigens. We added a few antigens from our expression libraries, 183 multi-ethnic, so Hispanics and African Americans and others, healthy controls. And another third study with now the best of those first studies, where we had another 101 patients. So altogether, we were looking at something like 300–400 SLE patients and 800 to 1,000 active and passive controls. Quite a busy table that gives you a ranking of the antibodies and antigens that we found in these 3 studies. giving you some p-values in the different studies, and we just concentrated on those that were highly significant in all or most of the studies. We see that there are some of the known ones like SSB or ribosomal proteins, the typical nuclear antigens. And we used an additional filter looking for prevalence. So how often does a patient or a patient cohort have this specific antigen? And then we can see ribosomal antigens in the first cohort, 35%, 25, 19%. So in the mid-20s to 30s, ribosomal proteins always about 40%. Some in differences and also in different ethnic groups. So that altogether made up a nice ranking where we said, okay, these are antigens we want to further evaluate. As an example, 6 of the best performing ones with receiver operator curves, AUCs of 80%, 72, 65, 80, 70%, quite nice. 3 of them are old school antigens, 3 of them are novel. If we combine them against healthy, we can reach 96%. Against different diseases, we always arrive over 85%. So looking at what literature says, what competitors do, we think that that's quite an attractive proposition. And we have, since we have our ISO certification 13485 since a couple of weeks, we have now started to develop bead-based tests remaining at the beads where we did the discovery with. And that's just a sketch of the different antigens we will be looking at. Now, the complexity of autoimmunity mandates us to not only look at SLE markers, but also to go for markers that are maybe not in SLE, like systemic sclerosis or CCP or any of the other known ones. By doing so, we also try to offer our skills and art to pharmaceutical companies, and we were lucky in a few cases where they did large screens with us. But most often they came back and said, we don't want to go for fishing expeditions. Our hierarchy doesn't like fishing expeditions. They have wasted too much money in that over the last couple of years. So we thought, based on our expertise now, can't we compose a kind of product type offer, which we did and launched this in May this year. It's called NavigAID SLE. So to kind of like a navigator on an ocean, try to find the right way. And also autoimmune disease, and aid means also help. So can we help people develop their drugs? Because what they face is similar to oncology, but maybe even worse. It's a mixed population of patients, and if you apply your drug in that mixed population, you will likely fail, as so many others did. If you have a stratification tool that can give you subgroups, homogeneous subgroups, here indicated by color, in SLE may be indicated by autoantibodies, your chances of being successful might be improved. And that's the challenge where we're looking at, and we wanted to solve 4 different questions that we repeatedly learned from pharmaceutical companies. One is, can we ensure enrollment of an appropriate SLE patient population? Now, that it seems to be a surprise, but in that complex disease, up to 20% of SLE patients in clinical trials probably do not have SLE. Now look, if you have 50% response and 20% are wrongly included, that is really a very, very pressing problem. Can we identify patients with high disease activity and or an interferon-1 inducible signature, which is one of the hallmarks in SLE? Can we diagnose associated organ damage, and can we stratify in different subgroups? So going back shortly, briefly, in the discovery, verification, validation study, we selected not only for diagnostic purposes, but now we selected for subgrouping of patients, autoantibody groups and cohorts, and we composed a sketch of a stratification array addressing those 4 questions. We added diagnostic markers for rheumatoid arthritis, CCP, and RA33. For systemic sclerosis, the 2 known ones, mixed connective tissue disease, polymyositis, Sjögren, and then SLE, and also in the other groups, disease activity, organ damage, and the interesting group of can we subgroup. That are some definitions I don't need to go in. Now that's the first question, differential diagnosis. We took systemic lupus, we took systemic sclerosis and a mix of other diseases and run them over our NavigAID SLE. And then we asked, based on their autoantibody profile, where are the similarities? So that's a similarity map. As soon as a patient has an autoantibody, it's given a 1. If it has not reached a threshold, it's given a 0. So you get, basically you create a 87 antigen line assay for every single patient. And then you sort these line assays by similarity. And those patients that have similar reactivities against the similar antigens are neighbors, and those that don't share antigens, they are not neighbors. And so we can see here is a red group. They share reactivities. The more orange it gets, the higher the reactivity number and reactivity score was. And based on the neighborhood, they also have the same antigens they are active against. In that region, you see patients that have low reactivity and they don't share these low reactivities. So with the bare eye, you can see one block here. Red is SLE. The next block is this is systemic sclerosis. And here you have other diseases clustered around. So a kind of differential diagnosis for a cohort is already possible at this stage. Having the interferon gene-related antigens also in that group of 87 antigens, we asked the question, if we only take antigens that are involved in interferon 1 gene regulation, how do patients cluster then? Now this is a fully different picture. Again, a similarity score. High interferon gene activity autoantibodies are clustered here. And then you have 2 more clusters with medium activity and 1 that has basically no activity at all. So from a standpoint of sorting of patients, you can do sorting. From a standpoint of, does it give any benefit to a pharmaceutical customer? It's now his interest to find out, is this group, has it good placebo response? Has this group good response to the drug, or is the response figure better here? And you can even go further down into 3 groups, good response, yes or no, good response, yes or no. And you could even go further in further groups, but then the market sizes for pharma gets too small. So you don't want to have an indication like SLE split down in 15 clusters. That's not good enough. That's too small. If you can have like 20%, 30%, 20% of patients and apply your phase 2, phase 3 trials there, you might be willing to do that. If you get smaller, it gets too fractal. What we also did, that we aligned disease activity, SLEDI, Systemic Lupus Disease Activity Index, indicated that by green and red, and we see, okay, high interferon signature autoantibodies is also high disease activity, and low also means low disease activity, so it seems to reflect biology. It's not only clustering by means of antibodies, it's also clustering by means of clinical picture. Now the last slide in that series gives you the segregation of SLE in homogeneous subgroups by utilizing all of our novel antigens that we found in our studies. You see that you have 2 big dividers, low reactivity and high reactivity, whereas every single branch here is 1 patient. Again, they were clustered with similarity, so brothers and sisters clustered together, uncles a little farther away, and wrong family even farther away. 1 patient is matched with himself at this line, so the diagonal gives you the per patient reactivity, and then you see, okay, here is a high per patient reactivity, which differentiates this group from that group. Here's also high reactivity, but the difference between these groups is they share their reactivities, so they are active against the same antigens, and they don't, which must be something biology wants to tell us. We don't know the reason for that, but we see that this is a common theme in SLE, which enables to separate that group out from that group. And now again, as a pharma company, you might find out that your drug works better here and even better there, because that's a phenotype that you can see. Interestingly, that bracket contains most of the patients that have a high interferon disease signature, as shown in the slide before. So with that, we believe that we can now answer those 4 questions. Can we ensure enrollment outlier detection is successfully done. If we spike in rheumatoid arthritis patients and others, it's easy to segregate them out of the SLE patients. Can we find patients with high disease activity? Yes. Associated organ damage, I didn't show data on that, but kidney involvement and so on we can show. And is it possible to stratify in different subgroups? Yeah, 5 to 35% of patient size subgroups can be found, and therefore we are kind of on air with that offer and have already had quite a number of interesting discussions with pharma companies, so we would expect that this offer is greeted with more enthusiasm compared to a 7,000 antigen fishing expedition. Conclusions. Omics discovery studies need high single data point quality. Omics discovery studies need high single data point quality. It's the most important part, and I fear the most neglected part in many of the studies. Based on the diversity, sample throughput of much more than 100 is relevant. If people come with small-scale studies, it's likely to be another filler for a black hole of lack of validation. High content is needed. Omics with 50 markers is not good enough. You probably need thousands of attributes to look at. And what we also think is quite important, seamless forward integration into suitable IVD technology is a strong plus. So if your discovery has many, many steps until you are at an assay system, you will lose so much of performance. It will be so expensive and time-consuming that you probably fail due to lack of funding on the way. Also, we believe in autoimmune diseases, many autoantibodies are yet to be discovered, and we will continue discovery studies. We have found about 50 novel markers for systemic sclerosis, of which 5 are now in CE-IVD development. We believe that multi-parameter tests, N is 10 to 20, might increasingly become part of clinical practice in kind of trusted plus new. The concern is reimbursement, whether diagnostics companies really go at such an early stage. If you can add 5% in sensitivity to a test, but those 5% are greeted with $3.50 per test, they're not going to do it. So for patients' sake, we need these 5%. For reimbursement campaigns, 5% are probably not good enough. So I'd like to conclude, yes, early diagnosis, differential diagnosis, biomarker support for drug development, companion diagnostics. You can bridge within 2 years omics-type discovery studies until you might be just shortly before reaching the market. If you have further questions, drop me an email, look at our homepage where we have lots of materials, and certainly I would be happy to answer any more questions. Thank you very much.